Hi, hi everyone. I hope I'm audible. Yes, okay. So let's get started. So today I'll be discussing about the phases of cell cycle. All right. So mainly we'll see what exactly is interphase and what is the M phase. So this is for class 10 biology. Okay. And the term 1 syllabus itself. Let's have a look at this. So before I start, let me introduce myself. I'm Vani Kari, biology educator at Unacademy. I'm an engineer with bachelor's and master's degree in biotechnology and a B.Ed degree. Also a founder of a startup where I grow microgreens by vertical farming technology. So join with us in Telegram for homework and class notes. You can join the channel Vani Kari ICSE Class 9 and 10 Biology and the Telegram channel of Unacademy ICSE and IC both. So this link you can find in the description box. So you can click on this link and join this channel for updates regarding the classes. So also we'll be conducting special classes on Unacademy platform. Okay, so all you need to do is download the Unacademy Learners app and start taking up these sessions, which are absolutely free. So you can learn from your favorite educator for free. So this is a free class. You as will be completing a certain percentage on these classes. The plus classes whole syllabus will be covered. All right, so engage with educators via poll. So this is basically will be giving a quiz, will be conducting a quiz where you can participate. You can know your rank, as will be sharing the leaderboard at the end of the quiz, so you can know your rank and also compete with your friends. Okay. All right, just a second. Yeah, so clear your doubts in the live special class. So we'll be conducting a classes, right? So separately there is a question tab where you can type in your question. You have an option to attach an image and send it to the educator during a live session. So they'll be getting a notification and will be clearing your doubt during the live session. Okay, so for the top special classes you can visit the link in the description. Now, Unacademy subscription features, you can learn live from the comfort of your home. So, main difference between the special class and these plus classes is that so you will have access to all the courses run by the different educators. Okay, so different educators will come up with their own courses for that particular month, so you will have access to all the courses once you take the subscription. So other than that, you can learn live from the comfort of your home. Then top educators of India are going to take the classes in one platform, that is Unacademy platform. Then there is regular doubt clearing session. So that is about after about every 3 session. The fourth session will be your doubt clearing session. Okay. So whatever doubts you have related to the topics that are previously covered, so you can clear your doubt in this session. So that is a doubt clearing session. Then complete syllabus will be covered. Mentorship and guidance is given. Study material, so we'll be uploading a PDF. So the same PDF you can download. So study material will be given in the form of a PDF format. And then there is practice test which is given. Live test series are given. The batch courses, here basically we have a English batch, we have a English batch. So once you take the subscription, so you will have access to English as well as the English batch. So this is completely your choice. You can go along with any of these batches depending on your comfort. Okay, then there is daily practice section, weekly mock test series, live quizzes will be there. Daily MCQ and subjective test series are also there. So this is about the Unacademy plus subscription pricing. So now since this is the very beginning, so since June is where the academic year starts, so basically you can go with the 12 months Or if you just want to try like how exactly this works, so maybe you can try with 3 months. So those who are planning for the further classes also, maybe you can go for the higher months of subscription. So this is depending on your convenience, you can go for any of these months of subscription. So if you are going for 12 months, so basically it is 17,250 for 12 months by application of the code Vani YT. So this is my code Vani YT, so where you will be getting 10% discount on the subscription. So you don't forget to mention the code here in the gift box. So there it's like, there you can see the cost that is getting reduced, that it is basically 10% discount that is getting reduced, and then you can go ahead with a subscription. Alright, so what we are going to discuss today, so this is about the cell cycle and the phases of the cell cycle. Any idea what is cell cycle? Any of you? Where do you see a cell cycle happening? No idea? Alright. Okay, so cell cycle is basically, a part of cell division. So now when we mention a cell division occurs, so before the cell division, so there is a lot of preparation that is required, because the DNA is getting divided. I mean the nucleus mainly, and the cytoplasm is getting divided. So for all that, double the amount of things are required. So for that reason, the cell cycle, it is just a preparation stage before, all the main divisions that occur. So under cell cycle only preparation stage also comes, and also the M phase. So that is what we have written. The phases of cell cycle is with respect to preparation stage. So that is mainly the interphase. Okay. So interphase when we mention, so this is basically a preparation stage just before the fertilization that occurs. Okay, so this is a preparation stage, and the other one being the M phase. Okay, so other one being M phase. So in that case, what exactly is M phase? So like, mitotic phase. So in that case, you will come across two different processes. So there is a karyokinesis, how the division occurs, and then there is a cytokinesis. So that is how the division of the cytoplasm. So this is with respect to cytoplasm, this is with respect to nuclear division. All right, so these are the different phase, like, different types that you will find in a M phase. So mainly we'll study about the interphase today and the M phase. Okay, so let's get started. So this is with respect to cell cycle. So let me just draw it here and explain you first. Alright. So now, let's imagine this one is the cell cycle. Alright. So here is the cell cycle. Okay. So cell cycle in center. So we'll take M phase only this much, because only about 5% is actually the M phase that occurs. So let's take only this as the M phase. Okay, so this one is basically the M phase that we are looking into, and the rest of it, so let's just take this part. So you see this part, so this is basically, 1 second. So you see this whole thing, right? So this whole thing is actually only about the 5% of the cell cycle. What is there? So 5% of the cell cycle is basically a M phase. Okay, so that is 95%. So biggest, which is the biggest one, 95% is basically a M phase. I mean, that is, sorry, the interphase. Okay, so here in this case, this whole thing, what do you see now? So this whole thing is actually a interphase. So this is about what I'm explaining here. So this is about the cell cycle that occurs. So in cell cycle, what do we see? So you see basically this one is with respect to cell cycle. All right, So in cell cycle, we come across the interphase, which is about 95%. So let's just complete this one. So this is basically a interface. This green part, what you see now, so this is completely a interface part where there are lot of changes that are going to occur. So we'll discuss all those changes that are going to occur in these phases. So when we mention about the interface, so since we already said that this is the preparatory phase, so you can see basically the G1 phase, which is also called as growth 1 phase. Okay, so then there is growth 2 phase, that you can S phase, and then growth 2. But some of the books have mentioned this to be a gap as well. So this is depending on your thing. So you can just mention it as a growth or a gap phase. All right, so this is completely your choice. So in that case, so there is a growth phase, that is G1 phase, then there is G2 phase, and you can see there is G1 phase, yes phase, sorry. So yes phase, and then there is G2 phase. So let's see what all happens here in these phases that are occurring in a cell cycle. Okay, so let's see this part. Now you see this part. One second. Okay, so this whole thing what you see now. Yeah, so I hope it is clear. So this is basically a interface. Okay, what is this? This is actually a interface where the interface is divided further. So this is divided here as G1 phase. Okay, so this one is a G1 phase. Then you can see there is a yes phase. So this is basically a yes phase. One second. So here let me just write the yes phase as well. So G1 phase, yes phase, and this one is a G2 phase. And under G2 phase, now this whole 5%, what is this? So this is about 5%. Okay, and this whole thing what you see, so this from here, let me just complete this one. So this whole thing is actually a interface itself. All right, so in that case, this whole interface, when we mention, just a second, yeah, so this whole interface what we mention, so this we call, yeah, one second, so this is basically about 95%. So this is about 5%, which is a yum phase here. So in yum phase, what exactly happens is there are two parts. One is cytokinesis and rest of it is karyokinesis. So let me just draw it here. So now this last part what you see. So I'll just write it here as a cytokinesis. Okay, so here you can see a cytokinesis that is occurring, and the yum phase is basically you can see different phases. So karyokinesis, so there is a prophase, metaphase, anaphase, telophase. So as such we have the phases here. So the first phase being in the karyokinesis, what you see, so there is a prophase. Okay, so first phase in the yum phase, that is mainly the karyokinesis, because this part what I have drawn here, so this whole part is of karyokinesis. Okay, so this is for a karyokinesis, and this part what you see, so this is for a cytokinesis. So under yum phase, so there is a karyokinesis and there is a cytokinesis. So first one being the prophase, the next being what, which is the next phase. So next phase is basically a metaphase. Okay, so there is a prophase, there is a metaphase, the next one being the anaphase. All right, so there is the anaphase, and then there is the telophase. So these are the phases that you can find in a cell cycle. All right, so cell cycle, mainly the G1 phase is there, yes phase is there, and then there is G2 phase. So let's see what all happens in these phases. Anybody has any idea what exactly happens in yum phase or G1 phase, yes phase, or a G2 phase? Any of you? No idea. So what exactly happens in G1 phase? So let's see this. Since we see this interphase, so this whole phase is actually an interphase here, which is about 95%. Okay, so in that case just a preparation is going on. So what kind of preparation? A preparation for the cell division. All right, so before the cell undergoes a division, so preparation for cell division is done. So that is done where? So that is done in the cell cycle, mainly the interphase. So now you know interphase is basically, you know, it is divided into three more phases further. So in that case we have a G1 phase, so we have a S phase, and then we have a G2 phase. So these are the phases that we can come across in an interphase, which is just a preparation for the cell division. So where exactly cell division occurs? So it is the M phase in the cell cycle. Okay, so cell cycle itself. Okay, so we'll be studying about the cell division as well, because this is the cell division that you study so much in detail. Okay, so that is all about, like, you know, you are studying prophase, metaphase, anaphase, telophase in detail. That is only about 5% you are studying in so much detail. Just imagine how much this 95 percentage of, you know, the interphase what we mentioned in a cell cycle that is happening. So it takes a lot of time. So in that case a lot of preparation has to be done before the cell undergoes division, before it undergoes for a karyokinesis, before it undergoes for a cytokinesis. A lot of changes will be done. So here the preparation for cell division, so that is the M phase, mitotic phase. So this one is divided into G1 phase, S phase, and G2 phase. So let's have a look at what exactly happens in a G1 phase. So here cell cycle, the same thing I explained. Okay, so there is a M phase, I mean interphase, and here is the M phase. So there is gap 1 phase, S phase, and gap 2 phase. Now here under the karyokinesis process, so this is for a karyokinesis process. So you can see prophase, metaphase, anaphase, and then there is a telophase. So only after telophase you can see there is a cytokinesis that occurs. Okay, so let's have a look at this. That is mainly with respect to the division of nucleus and what exactly happens in the G1 phase. So I'll just go on writing. Now the cell division. So what exactly, when exactly cell division required? So division of mature cell into two cells. So one cell is dividing into two cells. Okay. So this is a part of a cell cycle. So what is the heading of, like, you know, the title of this chapter? So cell cycle, cell division, structure of chromosome is there. Under cell cycle only comes the cell division. So that means it is a part of a cell cycle, whereas the cell division is just a 5%, and remaining one, that is, you know, the cell cycle, the remaining part is basically 95%, which is just a interphase. Okay, so interphase is a preparation phase, but the cell division phase is basically a young phase, which is just about 5%. So you can just imagine the 5% you are studying so much in detail, but there is a lot more happening here in this 95%, which you will be studying in higher classes. Okay, for now remember, I'll just tell you what all changes that are going to occur with respect to G1, yes, and G2 phase. So now cell division, when we mention, so this helps in cell growth. So when there is a cell growth required and replication that is required. So basically, let's say maybe you damage a certain part of your skin. Okay, so here, you know, the skin is damaged. So in that case what is going to happen, whenever there is a skin damage, so that old cell, or what we can say, a damaged cell has to be replaced. So in order to replace that old cell, or maybe we can say damaged cell, so you know, a cell has to undergo cell division. So before the cell which is here undergoes for the cell division, it needs to keep its DNA ready. So it needs to keep its DNA double the amount of DNA duplication. So DNA has to be doubled so that when this cell divides into two, so now equal amount of DNA has to be there in both the cells. So for that reason the DNA duplication is very important, or replication. So instead of a term duplication, we can also use the term replication, that is multiplying the amount of DNA that can be seen here in this case. Okay, so now distributes the replicated DNA to daughter nuclei. So that is what, so what exactly happens, the cell is there, so this is divided into two cells. All right, so that is before the cell division, and whatever DNA was multiplied here, so equal division of the nuclei will be done. So here some nuclei is given, that is genetic material, and here some genetic material is given. So it is not 70% genetic material to one cell, 50 30% genetic material to other cell. No, there is a equal distribution of genetic material, that is DNA. So if this is receiving 50%, so this is also is going to receive a 50% itself. So that means equal amount of DNA is distributed to the daughter nuclei. So now there are two methods where you can see a M phase occurring, that is cell division occurring. One is mitosis and the other one is meiosis. Any idea what is mitosis and meiosis? Just a second. No idea, right? Alright. So mitosis is basically, the cell division where all the body cells. So let's say there is a old body cell or a damaged body cell is there, or maybe it is infected by any pathogen. So we need to form a new cell. Okay. So that is with respect to body. So this is a completely body when we mention. So it is completely made of a cell. So any part in the body. So that can be of cell, and wherever there is a requirement for this cell to undergo cell division, as it is damaged or as it is affected by a bacteria or a virus, or as it can be old one, so in that case a replacement has to be done. So that is when a cell division occurs, that is to form a new cell. So with respect to that, the mitosis is for all the body cells. Okay, so complete body cells are undergoing a particular process, so whenever there is a requirement, so in that case they will be undergoing a mitotic division where the equal amount of DNA distribution that you can see. But meiosis, when we mention, so meiosis can occur, so let's say here, so only when the sperm production occurs, or maybe when the egg production occurs. Okay, so that is where you can see a meiosis occurring. So that is, meiosis is basically the production of, so this is basically, let's say parent cell was too young, that is diploid set of chromosomes were there, but after the meiosis is done, so whatever daughter cells are formed, so they just have a haploid set of chromosome, that is only one set of chromosome is there, where we call the meiosis as a reductional division. Okay, so this one is a reductional division, whereas the previous one, so that is a mitosis. So mitosis will do what? Mitosis is going to maintain the same numbers, like if the parental cell had 2n, so this also is going to have a 2n, that is two sets of a chromosome, or two sets of a DNA basically are there. So in that case we call this condition as a mitosis. Okay, so this one is a meiosis, this is a reductional division. Okay, so this is reductional and this is a equational. So why do we call it as a equational division? Because 2n is maintained as 2n itself, and hence we call this as a equational division, that is diploid is maintained as a diploid itself. Okay, so now let's have a look at the interphase, the phases during which the cell prepares for the cell division. So here in this case we have a different phases, there is a G1 phase, S phase and G2 phase. So I'll just draw and explain, just a second. Alright, So in that case, what do you come across? Like, so there is a phase during which cell prepares for the cell division. Then there is a phase between the two successive M phases. Then also it is called as the resting phase. So basically interphase can also be called as a resting phase. So cell growth and the DNA replica. So basically, replication. For DNA replication, for the cell growth, overall growth of a cell. So, a lot of changes that might be occurring. So, with respect to that, the interphase is one such phase where you can see a preparation is done in the cell body. So, before it goes for the cell division. Just a second. So, there is some network issue, I guess. Just a second. Let me just check this. No. All right. Okay, so let's have a look at the other one. So that is the gap 1 phase. So that is G1 phase. Okay. So the interval between the mitosis, what exactly happens in G1 phase? So this is about the G1 phase of the cell cycle. So which is basically mentioned as gap 1 phase, or we can also mention it as a growth 1 phase. Okay. So in this gap phase or a growth phase, so what exactly happens? So this is, all the raw materials are kept ready. Okay. So what are those raw materials? Let's say there is a requirement of, maybe we can say, the nucleotide requirement might be there. So that is including the sugars, the phosphates, the nitrogenous bases. Okay. So raw materials for the next, that is S phase. In S phase, the DNA duplication, that is replication occurs. So in that case, raw materials are required. So for that, as the raw materials like nucleotides, so some enzymes are generated here in this growth 1 phase. So this can be with respect to nucleotide. Okay, so this is basically including the what is this called, the phosphate group, the sugar group, then pentose sugar. So all these could be the raw materials that are required, and along with the enzymes. So let me just write here. So enzymes are also required. So now all this synthesis can be seen. So as you can see in this one, the interval between mitosis and initiation of the DNA replication. So here when you see, have a look at this picture. So I'll just draw it here. Okay. So now in this case, let me just show you this. So this is basically G1 phase is there. So what exactly do you see in a G1 phase? So there is, there are a lot of changes that are occurring. So mainly you can see the nucleotide, that is raw materials are kept ready here. So in this phase, what type of raw materials? Mainly the ones which are required for the synthesis of the DNA here, as the S phase is the one which is synthesizing the DNA in the S phase. Okay, so that means DNA duplication or DNA replication you can see here. So all that is required for the DNA replication is kept ready. So that we call as a raw materials are kept ready. So that is for which phase? That is for the S phase. So along with that, there is an increase in protein. So there is an increase in RNA content production. All right. So all these are there. So RNA is being produced, the protein is being produced, the raw materials are kept ready. So now once all that is done, so basically the DNA replication process is found. Just a second. So, yeah, I was explaining, but yeah, one second. Okay, so this is basically all the raw materials that are required. So these are kept ready here. So along with that, so some there is, as they are all kept ready, like nucleotide has to be kept ready, the enzymes. So basically here I was just writing in the previous one. here. Okay, so when an enzyme has to be kept ready, so that means it is a protein. Okay, so now for protein synthesis, what is required? So if a protein has to be synthesized, so there is a DNA which is already there, or there. Okay, so that DNA has to transcribe to form a RNA, and then this RNA has to get translate to form the protein. Okay, so in that case, as you can see, there is a RNA increase, there is a protein increase that is occurring. So as the conversion is occurring, so DNA to RNA and RNA to protein formation occurs, and hence in the gap 1 phase, or which is also mentioned growth 1 phase. So let me just write here. Okay, so the interval between mitosis and initiation of DNA replication, cell is going to grow continuously. Okay, then other than that, replication of few organelles. Yeah, this one I need to mention. So few organelles are getting replicated here, but the raw materials are mainly kept. Then this is also known as G1 phase. The cell in a metabolically active state. Why it is a metabolically active state? Because the raw materials are being kept ready. So that is like the enzymes, mainly the proteins, then nucleotides. So what are those nucleotides? So nucleotides, when we mention, the phosphate is required, then sugar is required, that is a pentose sugar is required, nitrogenous bases are required. So all these things have to be kept ready, and that's the reason. So this is a metabolically active one. Okay, so no DNA replication in this phase. So there is no DNA replication over here. So that is in the G1 phase, growth 1 phase. Okay, so the gap 1 also they can call, growth 1 also can be called, or this can even be called as a G1 phase only. Okay, so in the yes phase, what exactly happens? So that is the synthesis phase. So also this is known as the yes phase. So this is the phase where you can see a DNA that is being synthesized. So double the amount, whatever DNA was there in the cell, so that DNA is getting doubled here. Okay, so synthesis of a DNA, so synthesis phase we call, because synthesis of DNA can be seen as the DNA is being synthesized. So this we call as a DNA replication, DNA duplication. So DNA is being doubled there. Okay, no increase in the chromosome number, only the replication. So how is that possible? So let's say 46 chromosomes are there, so that is in cell. Okay, so in each cell, so this is in our cell, so 46 chromosomes that you can find. All right, so that means 23 pairs of chromosomes are there in a cell. All right, now what will happen? So usually when we draw the chromosome, so this is the structure of the chromosome that we draw. So here with the centromere in the center. Okay, so there is a centromere. So this one is basically a centromere that we show, and this one is basically a chromatid. So there is a chromatid. Now after you see the S phase has occurred, so that is mainly in the DNA replication has occurred. So we'll show one more arm as such. So we can show the chromosome like this. So why are we showing the double arms? Like, two chromatids are shown. So that is after the S phase. Okay, so that is because the chromosome number, so this is also one chromosome, but it is having only the double the amount of DNA. So that means chromosome have remained 46 chromosome only, but only thing is that whatever the content of DNA was there, so that has been double. So here double the content of DNA you can see. So that is because earlier this was the only strand. After the S phase here, so there is no increase in a chromosome number, but the replication has occurred. So you can see initial DNA amount was 2n. Okay, so initially what was the condition? 2n. So what is that 2n? So you can see this is 1 and this is chromatid that we are talking. Now final DNA is basically 4n. So that is 4 of the arms are being produced here, so as the double the amount of DNA is produced, but it is what, it is still a chromosome number 1, so we do not call it as 2 chromosomes are there, so this is only one chromosome that we can find. Okay, now initial chromosome number was also 2n, which is a 46. Now the final chromosome after the synthesis phase is done, so that is also a 2n itself, so that is also a 46 itself. Okay, so in that case, there is no increase in chromosome number, only the DNA content has increased. So moving on to the next phase, that is the gap 2 phase. Just a second. All right. So in gap 2 phase, what happens? So also this is known as the G2 phase, or we can also call it as a growth 2 phase. So some of the books have mentioned gap 2, growth 2, or a G2 phase. So anything is fine. So this is a phase. Now here I was just told, I was just telling that DNA replication occurs. So some of the histone proteins are also being produced here. So histone proteins are required, so histone proteins are also replicated here. And the next phase, that is mainly the G2 phase, so this is the preparation phase itself yet. So in that case, some of the organelles which were not multiplied earlier, so rest of the organelles are gonna multiply. So multiplication of the cell organelle you can see. Then there is synthesis of proteins that can be seen. So what is that synthesis of protein? As the cell organelles are getting multiplied, so what is the cell organelle membrane made of? So that is made of a protein itself, right? So there is a lipoprotein. So protein is required, lipid is required. So again there is an increase in RNA, again there is an increase in protein. As the RNA increases, the protein also increases, because DNA itself is converted to RNA. I mean, not converted, it is coded. Okay, so DNA is coding for the synthesis of a RNA, and RNA is coding for the synthesis of a protein, and hence whenever there is an increase in concentration of a RNA, so there is an increase in concentration of a protein that can be seen. Just check. Yeah, it is fine, but I don't know. Yeah, okay, fine, it is fine, all right. So in that case, there is a tubulin protein. Okay, so now this tubulin protein is actually required, so that is for the spindle fiber formation. So spindle fiber formation, when is it required? So spindle fiber formation is required, so that is during the cell division. So this is required when? So this is required during cell division. So basically when we mention an interphase, every required thing is kept ready here in the interphase. So that is maybe in the G1 phase, or in the G2 phase, or in the S phase. So basically all the things required for the cell division are kept ready here in the interphase, and easily the M phase can occur. And that's the reason, the interphase is about 95% of the cell cycle, whereas the other phase what you see, so that is the M phase. So M phase is not 95%, so what is the remaining one? So that is 5% is just the M phase, which is further divided as karyokinesis, and this is further divided as a cytokinesis. So there is a karyokinesis and cytokinesis that can be seen. Alright, so in that case, starts with the division of the nucleus, which we call as a karyokinesis. Alright, so why do we call this as a division of a nucleus? So that is because the DNA has been doubled in the S phase, that is the synthesis phase. As the DNA has been doubled, so you can see that the DNA is getting divided, so that is into two daughter cells. So in that case you can see karyo is basically a nucleus, kinesis is a division, so division of nucleus that can be seen here. So now karyokinesis occurs in 4 stages. So that is mainly the prophase is there. Then there is metaphase. Then you can see anaphase, and then there is telophase. So these are the 4 stages that you can come across in a karyokinesis. That is a division of nucleus. Okay. So this is with respect to what? This is with respect to division of the nucleus. Now this ends with the division of cytoplasm, so that we call as the cytokinesis. Alright, so there is cell cycle. So cell cycle, so there is G1 phase, cell growth, and cell grows, basically accumulates essential protein as DNA building block. Okay. Then next what we have, so this is the first phase, which takes about 6 to 12 hours. Then you can see synthesis of DNA, so that is cell starts replicate chromosomal DNA, result in pair of chromatid at centromere region. So this is basically yes phase is taking up to 6 to 8 hours. So this is taking up to 6 to 12 hours. This is taking up to 6 to 8 hours. Then you can see that there is a G2 phase that is there. So that is growth 2 phase. Final preparation for mitosis phase. So cell provide energy, that is ATP, and synthesize essential protein. So what is that essential protein? Like, the one which is required for the cell division, mainly the tubulin protein, which is required for the formation of spindle fiber, are being formed here. So that is, it will require about 3 to 4 hours, and the mitosis will just occur in 1 hour, so which is just a 5% of the whole cell cycle. So under mitosis we can see there is a division of nucleus, division of the cytoplasm. So one of the parent cell has been divided into two of the daughter cells. So this way the cycle is running. All right. Any doubt? So this is basically during cancer. Okay, so this is during cancer. So cells divide uncontrollably. So there is a division in cell that you can see, which is uncontrollable division. So why do we say that it is uncontrollable division? Any idea? So that is, they continuously undergo a division. Okay. Whenever a cell is dividing, so there is always a signal that will be initiated to stop the cell division at a particular process. Okay. But here what exactly happens is, there is no stopping. Okay. So there is no stopping uncontrollably, like, without any control. So there are no signal molecules to stop the cell division. So without any control, the cells will go on dividing. So they will undergo cell cycle repeatedly. So that will give that will give rise to a cancer cell. Okay. So during cancer cells, so basically the cells are going to divide uncontrollably. So chemotherapy is done. So this is to treat a cancer by killing the cells. So how is it done? So chemotherapy is the one. Okay. So some chemo drugs target different phases of the cell cycle. So what are, like, so there is one example which is mentioned. So gemcitabine. Okay. So it is a yes phase drug. So that means it is not allowing the replication of the DNA. So if the replication is not occurring, so there is no equal division of nucleus. So as the S phase itself is not occurring, so there is no further continuity with respect to the cell cycle. And that's the reason, the chemotherapy drugs mainly target the cycle, mainly the cell cycle, the different phases that are there. So those are targeted, and there is a blockage that can be done, like, prevention of a further stage or further phases prevented with respect to a chemotherapeutic drugs. Alright, so in yes phase of the cell cycle, so amount of DNA doubles in each cell. So this is with respect to amount of DNA doubles in each cell. Amount of DNA remains the same. Chromosome number is increased. Amount of DNA is reduced to half in each cell. So till now, like, just now I discussed the yes phase. When we mentioned it is double the amount of DNA, so it is the answer. Okay. So moving on to the next one. So in cell cycle, DNA replication takes place in, anybody here? Where exactly DNA replication occurs? No idea. Okay, so in cell cycle, DNA replication takes place here in the yes phase. So yes phase is where the DNA is getting double. So given below is a schematic breakup of the phases or stages of cell cycle. So which one of the following is correct indication of the stage or a phase in the cell cycle? So there is a karyokinesis. So whether C is a karyokinesis, wrong. So C is not a karyokinesis. D is synthesis phase. So what is this phase? There is a G1 phase, there is a yes phase, this is a G2 phase. Okay. And what is mentioned? D is synthesis phase is mentioned. So this is right. Then A is cytokinesis. No, this is a karyokinesis. So this also goes wrong. B is metaphase. No, because this is prophase. This is metaphase. This is anaphase and telophase. So B is metaphase also goes wrong. So this is the only option that is correct here. So that is D is synthesis phase. Okay. So DNA replication in bacteria occurs. So that is without the nucleolus, prior to fission, just before transcription during yes phase. DNA replication, so that is basically occurring prior to the fission. So that is the binary fission. Okay. Alright, so DNA replication takes place in yes phase. So this one is anyway repeated. So yes phase or a synthesis phase. Okay. Alright, so before I end this session, so let me just say about this. So there is a batch. So there is a gladiators batch for class 10, which is a all subject batch. And so if you are planning for class 10, so maybe you can go with the different batches that we have. So depending on your comfort, you can go for any of these batches. So if you are subscribing for class 10, so you will be getting class 11 and 12 subscription absolutely free along with this. And introducing community, so never miss on any update from your favorite educator. So get session notes and practice material. Share your feedback directly with the educator via polls. Check out the link to the educator community below, so that is in the description box. Okay. So Bucks Bounty, so this is opportunity for all learners to report any inappropriate content in the video. Be the first one to report a particular issue to claim your prize. Report any inappropriate content using the form in the description. Alright, so you can just check out these. Okay, so link you can find in the description box for all of this. So any doubts you guys have? Any of you? No doubt? Alright. Alright then, so subscribe to Unacademy, ICSE and ISC boards. Click on bell icon to get notified about these sessions. Download the Unacademy Learners app now. Like and share the video. And thank you all. So see you all in the next session. Bye bye everyone. Take care.